Hey, it's Dylan. I'm in charge of problem number 1 of the group problems, and it talks about a crossover. What is the allele and centromere composition of the 4 products of meiosis if there's a crossover between S and T? So this inversion already happened, and now we're going to see what happens if a crossover occurs between the S and T. So the products that you're going to get are the normal ones that we had, so the capital LMN through everything and the lowercase LMN through everything. And then you're also going to have the switches. So you're going to have a dicentric and an acentric. And so what happens is, as we can see, we follow along. Then once we hit the S, we follow the inversion loop that occurs. And so we go to the lowercase t. But because the strand is coming this way from it goes ST and this is ST, we have to include the rest. So it goes STUV, QPO, and then another centromere, then NML. And then the same thing occurs on the second part. So you're going to have capitalize XWVU and then T. And then once we get to that T, we're going to drop down for the crossover, and you see SRWX, and that's how you do it.
Hey guys, here we've got the week 8 group problems. Question 2. Non-disjunction in meiosis produces 50% aneuploid genes, whereas non-disjunction in mitosis can produce a mosaic phenotype. Meiosis in an individual who has suffered from a paracentric inversion produces a dicentric chromosome and an acentric chromosome. C. We have Turner syndrome is an example of a human disease caused by monosomy, while familial Down syndrome is an example of a disease caused by a Robertsonian translocation. So D here, we have the human liver cells can be 4N, 8N, or 16N. E, a terminal deletion in chromosome 5 causes Cruduchat syndrome, while an inversion in the X chromosome causes hemophilia. A mutation is a change in chromosome structure that can be potentially beneficial to an organism or an entire species. And G, when a translocation cross is formed, a linearity segregation pattern can produce viable gametes and offspring. All right, so question 3 here we have, how many total chromosomes would be found in humans that have the following conditions? So you got all those right there. Not too complicated, except for the familial Down syndrome. It actually deletes a 14 and it gains a 21, so you still have 46. B, which of the following terms is not associated with Charcot-Marie-Tooth disease? And so the only one that is duplication, because that's what it is. The rest of them don't really apply. Which of the following terms are associated with Klinefelter syndrome? And you have trisomy and non-disjunction. So D, which of the following terms are associated with type A hemophilia? It is deletion and duplication. Both of those can cause the irregularity on the X chromosome that causes type A hemophilia. All right, thanks guys.
Okay, this is James Leo. I'm doing questions 4 and 5 of lesson 8 group problems. So in group problem number 4, it gave us 2 alleles, homologous chromosomes, and one of them is DEFGH*IJKL, and the other one is MNOP*QRST. And then it also said that each of the chromosomes produce a different arrangement. So 4A asks us to diagram the synapses, and here it is, with the 4 different arrangements of the chromosomes. And 4B asks us to identify the allele composition of gametes produced by the alternate segregation. So here's the cross. In order to determine what that alternate segregation allele composition is, it's going to be this one and this one forming the first arrangement for the first cell, and then the other arrangement will be this and then this. So they're shown right here. The C asks us to identify the allele composition of the gametes produced by adjacent 1. So for adjacent 1, it's this one and this chromosome, and then this one and then this one, shown right here. And adjacent 2 for question D is these two chromosomes, and then these two chromosomes, as shown right here. So for question number 5, it asks us, it said that cats have 38 total chromosomes. How many total chromosomes would be found in the following type of cat? A is triploid cat, and that would just give us an additional, to get that, you have to divide 38 by 2 to find out how much there is in each allele, and then you times that by 3, that'll give us 57. And a cat that's monosomic for chromosome 12, that will give us 36. And then a cat that has suffered a terminal deletion on chromosome 7, it'll be the same, so 38. And then a cat that has suffered a Robertsonian translocation between chromosomes 17 and 18, it will also be 38 chromosomes. And lastly, a cat that has suffered a reciprocal translocation between chromosomes 6 and 9 will again also give us 38 chromosomes. So that's questions 4 and 5 of group problem number 8.